Hello and welcome. Now we'll talk about part two, bacterial cell culture, bacterial cell protein expression or the induction of expression, how to centrifuge or harvest the cells, and how to break the cells. So the overall workflow is just this. First, you have to transform the cells, which we covered in the last video, in the correct bacterial strain with all the various tricks to hopefully help you with the downflow work process. Now we want to take a single colony of this, put this into a liquid culture, let the bacteria grow for a bit so it gets a certain level of density early in a growth curve. And then we want to induce expression by adding in LAC-Z inducible promoter, use IPTG. Let that cell do its business. You're making more cells, making lots of protein in those cells, and eventually harvesting the cells, centrifuging them down, either storing or freezing them or breaking open the cells then and lysing, and then start the purification. Because we're talking about MDH and other MDH related proteins, we're talking about the histidine affinity tag as we talk about the purification. In the last video of this 3-video series, we'll talk about that purification and the downstream characterization in this. But for now, we have the clone in the plasmid. You've already transformed it in. Now we're going to talk about growing from a single colony or a group of colonies in liquid culture, spin them down, and lysing the cells. So you heard a little bit about antibiotics before. On the website, we have a bacterial culture PDF or protocol. It talks about culturing cells. It's important to know the antibiotic that you need to use is based on that plasmid. If it's a Pt2 vector, that means it's going to have a gene to break down and degrade the kanamycin. If it's a PQE60 Chigen vector that we use for the watermelon MDH, you need to use ampicillin. You're using some other the backbone, then you need to know the resistance gene for that backbone. It might be chloramphenicol, it might be streptomycin, it can even be tetracycline, and there's lots more beside this. Most of these are stored frozen in water or in ethanol for chloramphenicol and tetracycline at either 1000x or 500x fold concentration. 500x concentration for ampicillin happens to be 50 mg per mil. They're stored in the freezer and they're stored for a long time. And you can actually, in a pinch, you can freeze thaw these antibiotics. They're pretty stable for a long time. If you dilute out from 50 mg per mil to 1000 to 1x, you get about 100 mg per mil, 100 microgram rather per mil, which is 2 microliters of the 500x stock per milliliter of culture solution. You can see this table will walk you through there. Basically, we're talking about how to do an X factor or a dilution series. But different faculty or different researchers will have 1000x or 200x or 500x. So you always have to pay attention to the actual concentration of your stock, where it's stored, and know the final concentrations. And these are the standard final concentrations for ampicillin and kanamycin and the others. Culturing cells. It's always important to get the best expression to start from freshly transformed cells grown on a bacteria plate and then pick a single colony from there. If you have to, you can take bacteria that's been grown in a liquid solution, mixed with glycerol and frozen in a -80, chipping out a bit of that and putting it on a plate, spread it out, and then starting from that plate. If you really had to, you could pull out of that frozen glycerol, -80 stock of bacteria of transformed cells, and then throw that directly into a liquid overnight culture. That's the worst case scenario, the case you really don't want to do. But either way, it's always best we start this with a plated bacteria, an overnight small volume, and expand that into a large volume where you let the cells grow for a while, incubate with IPTG to induce the protein to be expressed. So expression, start with a clone, always make sure you have the right strain and the right antibiotic. It's best to pick a colony. It should always be the same, but in fact, if you pick 5 or 10 of these different colonies, some colonies will be better expressing than others. Some folks will take multiple colonies and throw them in the overnight culture because they figure if 1 or 2 of them are low expressors and the other, and 3 or 4 of them are high expressors. They'll all get mixed up anyway, and you want expression of protein. So different people will do, various ways of doing it. I tend to take 1 or 2 of them and then put them into a liquid culture for growth overnight. So you want to grow overnight, or if you have a problem with the bacteria and it's not growing, you never want to grow beyond 0.6, 0.7 units. So you might do a few hours in the daytime, then take that liquid starter, put it in the fridge till the next morning, and then expand there. There's lots of different tricks, but the standard way is to do an overnight culture with a small volume, let that grow till it gets nice and thick, and then take that and expand a portion of that into a larger volume. But again, there are some protocols that do not want you to go over 0.7, 0.6 OD because you start getting deleterious effects on the amount of protein being expressed. And so that happens if you have a troubled child clone that just does not want to express. So we culture in flasks. An important part to think about is you're culturing in these shaking incubators are standard incubating or standard flasks or flasks with baffles. The flasks with baffles allow for a better mixing and of your bacteria, so you don't get different layers of oxygen saturation. As your cells start growing, they're going to be using a lot of oxygen to do their metabolism. By mixing these, when they're open, you can ensure that the oxygen from the surface layer is getting diffused and mixed throughout the cell flask. You get a little bit better growth when you have baffles or little bumps around the sides of these things. Always important that this is loose or with cotton on it, so that way you can get air exchange, so the oxygen can get to the solution, but also important that you don't let just wild type microbes, bacteria, yeast, or whatever to get inside here. So we use loose caps or even cotton batting to allow air exchange. It's important, as it says here, to really not use more than half the volume, and actually it's better to even use a little less than that. The bigger the volume you get, the harder you get for mixing, and the less oxygenation of the lower tier of this bacteria. As you're swirling this way, you get the bottom end will be hypoxic, while the top will have the oxygen. So lower levels work best, okay? So let's show a couple of these examples. Now, what you'll see here is an example where a couple of students and I videotaped some things. And you'll see the process for doing this. And yes, there's no gloves. There should have been gloves, but to be honest with you, these bacteria are fairly safe. I'm more worried about contaminating my cells than these are safe bacteria to work with, but you should wear gloves. It's better PPE to wear gloves than what you see. So let's watch this. Going. All right, so we're going to start with by taking a little 70% ethanol or a 10% bleach solution, just clean off your work area before you start working with the bacteria, and you want to do that afterwards as well. What we're going to do today, we're going to take a plate that's already been transformed with cells. You can see on here that it says the name of the plasmid. In this case, it's human malate dehydrogenase 2. And it says T224D. That tells us that the threonine in the 224th position has been mutated to a D, or an aspartic acid. This is in a cell line called BL21DE3. It's always important you tell the clone so you understand the backbone and the strain of the bacteria and when it was produced. You can see there are nice individual colonies on this. So what we want to do is pick one colony and put it in a small volume so we can culture that and then expand it to make protein. Is to, again, take a colony, a single, I'll edit out later, mound of cells, and transfer that into a liquid volume. Maybe a small volume or a larger volume. There are two basic kinds of vessels that we use here. We use test tubes and we use flasks. The difference between the flask you see here and say this flask is in the lip and in the bottom. This has a straight top lip, so you can put a cap like this. This cap is vented and loose to allow air exchange. They won't fit on this. Not enough so that way it wouldn't fall off when you're doing it. Larger vessels, same kind of thing. Have their own caps.
You notice with all three of these caps, we have some sort of a top that we have a little bit of room for air to exchange. That's very important. If you had a vessel like this and you didn't have a cap or couldn't find a cap, cotton batting is often used to stuff in there. Again, enough so it won't fall in. That allows air exchange, limits gross contaminants from getting inside your solution. What we're going to do is we're going to take a LB media, a Lyria broth media, that's already been added with kanamycin, which is the antibiotic of choice for the backbone of the plasmid that we're using. We want to take one of these and put it into a small volume here. We're also going to do this to a larger volume just so you can kind of get a feel for what this would look like. Depending upon what your ultimate goal is for expression volume, you may need to have just a small one or a large starter volume. So these are called starters. We'll let these culture overnight. They'll get nice and dense with the cells. And in the morning, we'll expand them into larger volume. So a large volume like this would be expanded into this. A small one like this might go into a vessel of this size for a 10 or so, 50 mil or 100 mil culture. So we'll just transfer, in this case, $50 mils of antibiotic or LB media containing antibiotic into this vessel. Again, we're just seeding the fingers from the cap and from the tin foil so it remains as sterile as possible. If we're not in a sterile environment, the kanamycin helps. All right, so I've transferred 50 mils in this vial. Notice we take a very small total volume of this because it's going to shake. We want this to shake nicely, but just so it can get aerated, but not so filled that it will overspill. Both of these flasks work fine. The difference is these bafflings here helps keep the bacteria culture in suspension a little bit better than this. Both will work. If you have tough growing cultures, sometimes the specialized baffle columns or flasks are better to use. So now what we want to do is isolate one of these colonies and inoculate our media. So we're going to just take a single isolated colony. Here we have colonies all next to each other. They're all producing an enzyme that will help degrade the kanamycin. We want a single isolated colony. We'll just scrape the surface. And then in the solution, rinse it off. We'll do this again in another way. Sometimes take a pipette tip and find an isolated colony. Just scrape that colony up. And you don't need to get all of it. Place it in the cap. I like to just leave the tip in there. And then that. We're done. Wrap this back up with parafilm. Either have a loose cap like this so the air can exchange, or a cap like this so air can exchange, or use cotton in either one of these. All right, so we're going to start with by taking a little 70% ethanol.
All right, so what we do next is take those starter cultures and put them in a shaker. So here's some specific information and safety about using the shaker incubators.
All right, so this is a shaking incubator. We typically keep this at 37 degrees. For tubes like this, you want less than half, a little bit better than most. This is 5 mils in this tube. Simply put them in this holder here, so it's set at an angle that keeps the liquid moving and aerated. And then find one that fits. See how this doesn't fit? That's not the right size, so we need to find the appropriate size for that.
Let's say that the folks who use tape and other things to make it fit, don't do that. You're just asking for a mess to happen.
All right, so this is a shaking incubator. All right.
So now you've got your cultures going. You started from a plate, you've gone overnight, and it's growing here. Then ultimately, you're going to take a portion of this and expand that to a large culture. Let that grow for a certain period of time. Get the cells early in the growth curve in an early density, and then induce the cells. The more cells you have, or the bigger culture you're inoculating, the more you need to start with. Starting volumes about 50 mils. If you're going to end up culturing 50 mils, use about 1 mil of a 10 mil culture. If you're doing 250 mils, 5 mils of a 10 mil culture will work. Doing 1 or 1.5 or even 2 liter cultures, 20 to 40 or even 50 mils of a starter culture is a good start. You could start even with only 1 mil of a 10 mil culture, but then you've got very few cells and it takes a long time to get to the growth density. And we measure that density, you'll see in a second, with the spectrophotometer looking at the optical density of about 0.4, 0.5, maybe even 0.6 when you induce. If you have a very small amount of culture in a large expansion, it's going to take a long time for that to get there. So with these values growing at 37 with reasonably healthy cells, it should take 1 hour to 2 hours or so before it's time to induce. Once you've done that, you're going to monitor, and we'll show you how to do that in a moment here, at 600 nanometers. At 600 nanometers, what you're really looking at is the light being scattered. It's looking at the density of the cells. You want to do this initially so you can get a handle on what the readings are absorbance, but they're more properly called optical density because you're not actually looking at absorbance, you're looking at light scattering. And then you want to keep doing this and figure out about E. coli double about every 20 minutes. It doesn't mean you're doubling the absorbance or the optical density, but close to that. So once you get started, after about 1 to 2 hours, depending on how everything you've done, that optical density or the OD 600 will get to be about 0.6. That's when I like to induce with IPTG. If you have problems with cells where they stop growing because the protein you're making is deleterious to the cell, maybe you want to get a higher density before you add IPTG. So you have lots of cells making some protein versus a modest amount of cells growing and dividing and making protein. So there's different tricks to this. Then once you do that, you're going to standard wise add the IPTG at an OD of about 6.6 or so. Let it go at 37 degrees for 4, 6, or 8 hours. Centrifuge, and then you can store the pellet or start processing. If you need to, if you have problems with too much protein being expressed, or even with low expression because of inclusion bodies, oftentimes we'll decrease the temperature to overnight at room temperature, 20 degrees. We've even done 16 degrees where the bacteria grows really slow and produces protein very slowly, but you get a lot more total protein by doing that if you have low expression or expression problem plasmids. And so by going 24 or even 48 hours at 16 degrees, 24 hours or overnight, 12, 15, 16 hours at 20 degrees, you can, by decreasing that temperature, you can get pretty good expression. But if you have a strong expressor, there's no need to that 4 to 6 or 8 hours of induction at 37 degrees, then harvest cells by centrifugation. So on the website, we have the protocol for expression and expansion. You start with the expansion of the starter culture, what we talked about, and you want to get the OD to about 0.5, 0.6, maybe a little bit higher. Then you're going to want to take that expansion and induce it either at room temperature or 37 degrees. Alternatively, a third option would be at 16 degrees. For your standard expression, follow the 37, where you go 4, 6, or even 8 hours at 37 degrees with shaking. After you've added the IPTG, then you can spin down and freeze. Or as I said, for proteins that don't express well or over express into an inclusion bodies, and you just don't see a lot of expression, we then typically go overnight, 14, 16, or even 24 hours. For real difficult problems, we'll do 16 degrees for about 48 hours. So let's watch some of this.
All right, so we've let those go overnight, and now we're going to transfer them or inoculate, expand the culture, so then we can let it grow.
He should be wearing gloves, but he is wiping his hands with ethanol in between. He should be wearing gloves.
and then induce. Again, we'll clean up our work area. We're going to do this in 2 batches. We're going to transfer 1 ml of this into 100 ml of fresh culture, again containing kanamycin, and we're going to take the 20 ml that we started and put it into 1.5 liters. Now, here, this is pushing the limit. We really should only have about 1 liter in this, but we're going to push it for today. If we were doing the exact best culture, we know this will grow pretty well in these conditions. We should really split this into 2 larger vessels. 100 ml in a 250 ml flask is certainly good. Notice this one doesn't have the baffles. Again, that'll grow just fine. You have to use what you have available to you. So now in this case, we just simply transfer 20 ml from here. There. Flip it. Name on there. And we'll take 1 ml with culture. And see this liquid culture. Those with the old culture, rinse it in bleach, 10% bleach, rinse it with distilled water, and then wash your glassware as normal.
All right, let's continue on. Now we've seeded with our starter and we're expanding. Not yet adding IPTG.
Let these incubate in culture at 37 degrees. These clones we know like to express better at colder temperatures for a longer period of time. So we'll start the pre-induction culture at 37, then we will cool them down to 20 degrees and then induce. This one's already fairly dense because we seeded with a large inoculant. So this won't have to culture for very long until it hits an optical density of about 0.6. This is probably looking at about a 0.4, 0.5 right now. This one's got a little bit longer to go. And again, it's just got to grow enough to catch up. There we go. Making sure that your flask fits securely and snugly, not loose and wobbly. Those start. All right, so we let those go over.
All right, so depending upon the lab you're working in here at USD, we have the verniers, and sometimes they'll be set up near the shakers. Other times you may have to go back to the lab. So for those who have verniers, here's how you can start checking your optical density using the vernier spectrophotometers. All right, so depending upon your lab, you're going to want to be able to check the absorbance, or rather the optical density, how thick your culture is. We want to induce these early in the log phase. So we usually wait for and measure the optical density at 600 nanometers. Starting as a blank using LB with no culture growing in it. Oftentimes we keep one of these short-term covered in parafilm in the cold room or the refrigerator. You want to make sure if that's the case that you don't have condensation impacting your zero. If that's the case, let it warm up before you use it and make sure you wipe off the edge. When using semi-micro cuvette like this, you're looking for the 1 centimeter pathway. And in this case, using the vernier spec, you can see where the light source is and the detector is based on the arrow. Want to make sure your 1 centimeter pathway is in this orientation, not here. You would have either, if it's a frosted cuvette or in this case it's clear, you would have less than 1 centimeter, and that would mess up your calculations using Beer's law. So we'll start by waking up the spectrophotometer. In this case, we've already started it, but we'll go to a mode as a time base. For this one, it's nice and simple. If you don't have it on time base, maybe it's on full spectrum, you have to just select, in this case, the time base, hit OK.
Assuming that somebody else had it at a different wavelength, you click in the red area, hit change wavelength, and hit 600. This spectrophotometer will put you to 599.6 based on its grading and filter sets. Then we'll hit this again and tell it to calibrate. It takes the dark sample, and you make sure that there's nothing inside here, although it just shuts the shutter, so I don't think it really matters. This instrument has been on for about an hour, so we don't need to let it warm up for another 70, 80 seconds, so I can skip warm up. You want to let the instrument up and the bulb to warm up if it's been cold. So as it says, you place your cuvette in the device. This is your blank cuvette with your blank medium. Hit finish calibration. It told you it took 24 milliseconds for its sample time to do that calibration. Hit OK. And now we're set. In fact, you don't need to do anything else. If you leave this on at that 0, simply put in your new cuvette with about halfway filled. The light path, this instrument reads is sitting about here. So you want to be about this high with your solution. The light path will be a little beam of light going through the sample here, right about here. So you want to fill it up about halfway. All right. So if you keep this here, this 0, as long as it's still clean, you can always double check. Your absorbance should be 0. You do the same thing with some of your culture. Put it in, look at your absorbance. Then you'll know when it's time to induce. Okay, so the cultures have been incubating. They've been cooled down to 20 degrees because we're going to induce these for overnight at 20 degrees. At this point, you can see that they're cloudy, so there's turbidity, light's being scattered, and we're going to want to measure that as optical density, not absorbance. We're not looking at how much light is absorbed. We're looking how the dense this is optically. It scatters the light, how much light does or doesn't get past the bacteria suspension culture. When you do this, you typically want an OD6 at 600 nanometers to be about 0.6. If you go too low or too high, the cells aren't dense enough or they're too dense, and you won't get the kind of protein expression you're hoping for. We'll just take, again, here we have the cuvette. It's about twice as high as it needs to be. Notice it's a square instead of a semi-micro cuvette. That's fine as long as, again, your 1 centimeter pathway is in the right area. Put it there. And we can see we're a little low here. So we're going to let this one culture for about another half an hour to an hour till we get about a 0.6 OD. Now let's check our bigger culture. Again, when we're done with this, all the plasticware will be rinsed with bleach. This one's a little less dense, so this has got to go for a couple more hours.
And so each time you do this, that's what you do. You watch the OD when you get 5.6, maybe 0.7, add the IPTG, and then based on your experience or your instructions, you let that go for 6 hours, maybe 8 hours at 37 degrees, overnight at 20 degrees or longer at other temperatures.
Okay, so.
All right, so we're inducing. So what does induction really mean? All we're doing is, if it's the PET expression system, then we're adding IPTG to work with the lac operon to drive the expression of a protein, okay? And instead of lactose to drive this, we use IPTG. IPTG is an analog, but with this group here, instead of lactose, it doesn't get metabolized by the enzymes of the cell. So this can constantly bind to the lac operon, relieve this so we can get the nice expression. We can take care of that kind of nice leaky issue. So now it's time to talk about cell harvesting. All right. So if you've ever done a centrifuge, these are both very expensive and dangerous and easy to mess up. The bottom line is you want to make sure that your tubes that you're using in the rotor are the appropriate tubes, that they fit the tubes into the right speed. If you're not sure, talk to your instructor. don't guess. If you use a conical tube and it's a round rotor, you're going to distort the conical tube, they'll break, then you get an unbalance. And this is not your mom's washing or dad's washing machine where it just gets out of balance and stops. You can actually break the rotor, break the spindle, cause tens of thousands of dollars of damage and maybe even personal harm. This can be done in even liter or 2 liter volumes. As you see here, there's a nice huge cell pellet there. Ultimately, what you want to get is a nice tight pellet, right, that you see here, and the LB media to be separated out. So let's talk about centrifugation, or let's watch the video. All right, so we've cultured these overnight at 20 degrees. You can see a nice thick, dense culture. We have both our 100 mil culture and our 1.5 liter culture. Everything is going really good. So now what we're going to do is we're going to take the 1.5 liter culture and split that into these two 500 mil vials and centrifuge them down. There will be a little bit of remainder, so we'll pour out the resulting supernate and then add the remaining culture evenly into these two spin again. So we'll have the 2 pelleted cells into these two. For the 100 mil culture, it's just simple to do into the 50 mil falcon tubes or conical tubes like this and store them either for immediate lysis or later on for by putting them in the freezer. We can take them off, thumb out, and use them out later on. All right, so now we've filled these and we need to balance them. You can either balance them on a digital balance or you can balance them on a pen balance like this, which our lovely assistant, Nate, hi Nate, is going to do. Go ahead, Nate. We kind of already cheated here. We already know that they're balanced. If they weren't, use a smaller one, put them in there, keep the caps on each balance, and then use a pipetter to even them out. So that one's good to go. Now for this, we're going to centrifuge these down again into these two. Now, for these tubes, we can use a simple tabletop centrifuge like this. All right, door, stop, pause, all that good stuff. When you want to centrifuge, you want to make sure that you have equal mass across from the center. So one goes here, one goes here. All right. If you have multiple tubes, you have to make sure they're balanced across from them. So if you have 2 tubes, or 4 tubes, they could be 2 tubes of one weight or one mass and 2 other tubes, there we go, 2 other tubes of a different mass. And as long as they're balanced across from each other, not this half versus this half, then you're good to go. For centrifuging bacteria like this, 3,500 RPMs, or correction, 3,500, stop. So when centrifuging bacteria cells like this, the centrifugation force or relative G force of about 3,000 to 4,000 is all you need. 3,500 times G is about right. You do not need this. You do not set this at 3,500 RPMs. The distance from the center to the actual tube for each rotor can translate out to different G forces. So it's RPM does not equal G force. Make sure you undergo centrifuge training before you do this. For these tubes, we will just use a different centrifuge because the rotors for those tubes don't fit in the tabletop. But there's no problem for this. Again, we'll look at the rotor, we'll look at the instructions, find out how fast, how many RPMs to get 3,500 times G. For 10, 15 minutes, we'll take care of it, and then we'll have a nice pellet. All right, so now we've spun down the cells, and we have this cell pellet. Some people take that pellet and combine all the various tubes of the same prep into one tube, freeze that in a falcon tube or some other kind of tube. Some folks will scrape that out and then put that in even a Ziploc baggie and then put that in the freezer with just the paste. And that way you can take that out frozen, open that, and then start from there. So you can store these cell pellets in the freezer, and that works out really well. So now it comes time to break your cells. Depending upon what you're doing, there's different ways to do cell lysis, and there's just not enough time here to go through all of them. So we're going to talk primarily about something called XJ autolysis cells. So these are another specialized BL21 strain from a company called Zymo Research. What they have is in these BL21 cells, in addition to whatever plasmid you placed in there, there's already a second gene under an arabinose plasmid driving the expression of a protein called bacteriophage gamma endolysin. And this endolysin will then break down, degrade the bacteria cell wall. And what that means is if you take the cells that are expressing endolysin and your protein, they'll be okay until you freeze thaw them. And then the expansion of the liquids when they freeze, water when they freezes, will cause the cells to rupture if you go through 1 or 2 cell cycles. And this is actually really efficient without any specialized tools. So folks that don't have a sonnicator or some other, a French press or something like that, using these autolysis cells with this endolysin gene in them is a very efficient way to break the cells. Otherwise, what a lot of folks will use is a combination of an enzyme called lysozyme that degrades the cell wall, and then they still sonnicate, and they often don't use detergent. Some folks will call Triton X100. We try to stay away from that because we get good cell lysis or breakage without the addition of that detergent. When you do this, you want to make sure you're adding PMSF. So that way PMSF is a serine protease inhibitor. So as you break open any cell, the natural proteases of the cell are now exposed, and they'll find proteins, including the one you're trying to purify, and start cutting those up. By having PMSF or a series of other protease inhibitors, you can slow down that protease. PMSF is pretty good for a number of the serine proteases found in bacteria cells. And for MDH, it's pretty stable. And so while using a cocktail, a much more expensive mixture of proteases helps, PMSF alone in that first lysis step is good enough. Often we use a little bit of beta-mercaptan ethanol to keep the redox state so the proteins stay folded and native more as they break out of this, as the cells are being broken. With larger preps, larger than 15, 20, or maybe even 100 mils, you have so much genomic DNA in there as you're breaking open these cells. The DNA unwraps from the histones and other proteins and becomes really viscous, especially in large half-liter, 2-liter preps. And it's this thick, like solid goop, like somebody blew snot in them, actually. And so sometimes the sonnication alone will take care of and degrade all that DNA, break it into smaller chunks so it's not so viscous. Other times you may need to add DNase A, an enzyme that will then start breaking down or restriction digesting up that DNA, so that way you lose that viscosity. Because if it's viscous like that, the cells won't, the centrifugation will end up with really viscous, unclarified samples. And so once you do the centrifugation, you want a high-speed centrifugation so you can get 2 phases. You can have the proteins that are soluble in the leftover solution, that's considered the lysate, and the protein pellet. Let's walk through how using a sonnicator, and this is very specific for this kind of sonnicator. Basically, we want to turn this on, be very careful with the probe, never put the probe on, as we'll see in the video here, until it's the sample is in, or the probe is in the sample. You want to turn this, start low and turn this up high enough so you get good sonnication, but not so much it gets foaming or what's called cavitation. So let's watch.
Start low, adjust by turning up and down, on and off here. So there's the tip in here, so it's suspended. Do not touch the tip to the side. And then increase the power.
So how much and how long and the power that you use for this depends on the shape of the vessel, the depth and density of that sample in the vessel, and actually the location of the sample. So you really do, you can't say X power unless you always have the same volume and the same kind of density in the same shape vial. So I literally start low, turn it up till I get a really robust, you can feel it, you can hear it, you can see it, sonnication. Let's see what that looks like here. Here you see the sonnicator going to town. It's breaking up the cells. I shut it off before I remove the solution from the tip. And any faster than that, you get what's called cavitation. It would just get foaming, and then the energy doesn't get into the liquid. It actually goes into the air interface. And so if you get the foaming, that's fine. Shut off the sonicator, let the sample sit for a few minutes. Now I'm doing this here so you can see this with my hand holding this. You really should have this on the ice. This sonication with the vibration and rotational energy that's shot into the solution will get it very warm and heat, and then you start denaturing your protein. So this really should be done on ice. I did this here as a demonstration model to show you what it would look like and sound like. But again, you take the ice bucket, raise it up, and lower the probe down into the sample.
Or so that the pellet is on the.
So now in this case, I've centrifuged that lysate at a high rate, not the same 3,500 times G, much higher. I'll look at your protocol to see that and see what your instructor has for the appropriate tubes and centrifuges. But after centrifugation, you need to be very careful on how you're separating out your pellet from your lysate. You want to minimize the amount of pellet that disturbs back into that lysate. You want a very clear, not cloudy solution. And clear meaning not colored, but that there's no particulate matter in there. Otherwise, it'll get in the way of the purification.
Start low.
Well, that's not who we wanted to hear.
Bottom and pour over the pellet like this, minimizing the disruption, leaving the pellet intact. If you pour this way, a loose pellet can fall into your solution, redistributing and contaminating your sample. Notice the clarity. This is ready for a column. If it was cloudy, it's because there's some pellet left inside here. It needs to be respun. Never apply this lysate to a column if it's cloudy. It needs to be clear. A color from the bacterial lysate's fine, but it needs to be clear, cannot have particulate matter making it cloudy.
All right, so that'll be the end of part 2. Next will be part 3, where we talk about the actual purification and characterization. Thank you.